everyone, in this part of the lecture I will be talking about bioactive molecules. This include organic acid, alcohols, enzyme, drugs like cyclosporin A and statins. So all these chemicals, they are produced by microbes. Now bioactive molecules, they are chemicals which influence functioning of living system. They affect living tissues. So let's see definition of bioactive molecules. They are substances which have an effect or elect response from living tissue. They include all those chemicals which influence functioning of living system. Now in this category, first of all, I will be dealing with enzymes, and in enzyme, let's begin with pectinases. Now, as the term suggests, pectinases, they are involved in breakdown of pectin fibers. These enzymes, they are obtained from Aspergillus niger and Bisoclamis fulvo. Now, where these enzymes, they are used? If you form fruit juices at home, you see that these juices, they are thick. Why they are thick? Due to presence of fibers, pectin fibers, protein fibers. But if you buy fruit juices from market like real fruit juice, Tropicana fruit juice, they are relatively clear because they don't have fibers. Digestion, clearing of fibers is done with the help of pectinases. So this is one important function associated with these enzymes. Clearing of fruit juices. Second, retting of fibers. Retting of fibers means separation of fibers from stem or other parts of plant. Along with that, they are used for fermentation of green coffee and enhanced juice extraction. Amongst all this, you have to focus on the significance, on the use which is given in the NCERT, that is clearing of fruit juices. Moving on to the next one. Proteases, they are involved in the breakdown of protein fibers obtained from Bacillus subtilis, Mortella, Aspergillus oryzae and Penicillium species like Rock 40. Now protein fibers, they are used in detergents. They are used for making detergents. In detergents, they are used for cleaning protein-based stain. Also, they are used for clearing juices. In juices, they are used for digesting protein fibers. So, these are two important significances associated with proteases. Lipases, yes, they are involved in digestion of lipid fibers obtained from 3 microorganisms. And to learn names of these microorganisms, you can use this mnemonic here, that is CAM. One is Candida lipolytica, Aspergillus niger, Mucor javanicus. Now, lipases, again, they are used for making detergents, Tide, Ariel. They have lipases, and in detergents, they are used for dissolving lipid-based stain, dissolving oil-based stains. Now, amylases. Amylases, these are enzymes which break down starch into maltose. Maltose is a disaccharide. It is a sugar which is a disaccharide. It consists of 2 glucose. Now they are obtained from Aspergillus species, Rhizopus species and Bacillus subtilis. So these 3, they are fungi and this one is bacteria. Now what is the function? Amylases, they convert starch into sugar. So surely they are going to result in sweetening of bread. Along with that, they are also used for clearing starch-related stains in utensils and clothes. Streptokinase, this is very important enzyme. Now, streptokinase is one enzyme which is obtained from a bacterium. Name of that bacterium is Streptococcus. It belongs to Actinomycetes. Now, this is a clot buster. When we say clot buster, it means it helps in dissolving blood clot. For that, we must know how blood clot is formed. Now, fibrinogen, it is changed into fibrin. Yes, fibers are formed. These fibers, they entangled RBC, which results in the formation of blood clot. Now, streptokinase, it has ability to break fibrin. Now, breakage of fibrin will result in dissolving of blood clot. So it means you are going to say that fibers are dissolved. Lysis of fiber is done by streptokinase. That's why you are going to say that it has fibrinolytic effect. Yes. So streptokinase is obtained from Streptococcus. Genetically modified Streptococcus is used for forming streptokinase, and this streptokinase is going to work like important blood clot buster in humans. So it is a blood clot buster. It can break fibrin. That's why you are going to say it has fibrinolytic effect. It is used very well to resolve blood clots in vessels of patients having heart attack. Now, next one, cyclosporin A. Now, this chemical, this drug is obtained from Trichoderma polyspora and Tolypocladium inflatum. Both of them, they are fungi. So, cyclosporin A is a product which is obtained from fungi. Now, what is its significance? It is an immunosuppressant. It will not allow immune system to work. It is used to suppress immune system in those organisms where organ transplantation has been done, so that it will not allow rejection of transplanted organ. Now, in our body, there are two types of lymphocytes. That is B lymphocyte and T lymphocytes, which are actively involved in immune responses. And you have to keep this in mind that cyclosporin A, it is going to inhibit activation of T cell response. Yes, it has triple role. Yes, cyclosporin A has triple role. One I have discussed here, that is, it is a immunosuppressant. Along with that, it has antifungal properties and anti-inflammatory nature. So, second one, antifungal and anti-inflammatory. Right? Statins, these are drugs which are used for lowering cholesterol. So first of all, we have to see how cholesterol is formed in human body. For forming cholesterol, this substrate is used, that is malonate. Malonate is acted upon by this enzyme, that is HMG coenzyme A reductase. Full form is beta hydroxy beta methyl glutaryl coenzyme A reductase. Yes, it is malonate. With the help of this enzyme will result in the formation of cholesterol. But how statins they are going to act? Statins, they show structural similarity with malonate. If they show structural similarity with malonate, it means they compete with malonate for active site present on this enzyme. If statins, they will join with this active site. So this is going to inhibit this reaction. What you are going to call such type of inhibition has, this is known as competitive inhibition. So competitive inhibition inhibits synthesis of cholesterol, and that competitive inhibitor is statin. So statin, first you have to keep this in mind, it is obtained from a yeast. Name of that yeast is Monascus purpureus. It is blood cholesterol lowering agent. Yes, here I have mentioned this chemical which forms cholesterol in human body, that is malonate, and this enzyme activity is very much essential for cholesterol formation, and it is going to be inhibited by statins. Now we are going to talk about organic acids which are formed by microbes. Aspergillus niger, this fungus is involved in the formation of citric acid. Now citric acid is used for dyeing, inks, medicines, flavoring and preservation of food. Citric acid is very well used in preservation of jams, jellies. Acetobacter aceti, this bacterium is used for the formation of acetic acid, and acetic acid is present in vinegar. Yes, 13% acetic acid is present in vinegar. So it is used for vinegar preparation. Clostridium botulicum, yes, it forms butyric acid and it is used for making rancid butter. And how can we forget lactobacillus, which forms lactic acid, and this lactic acid is used for curd formation. Last one. Aspergillus niger and penicillium, they form gluconic acid. This gluconic acid is very important because it is source of calcium for infants, cow and lactating mothers. So this is all about bioactive molecules. So now let's take a few questions here. Organic acids used in preparation of vinegar is acetic acid, and this is formed by bacterium Acetobacter aceti. So right answer to this question is option number 1. Statins are competitive inhibitor of. Statins, they helps in lowering down of cholesterol by inhibiting enzyme, and name of that enzyme is hydroxymethyl glutaryl coenzyme A reductase. So our right answer here is option number 2. So in this lecture, I have talked about role of microbes in forming household products, then industrial products, and at the end, I have taught you role of microbes in formation of bioactive molecules. Now, in the next part of the lecture, I will be talking about role of microbes in treating waste, in treating sewage. Yes, we are going to discuss sewage treatment plant in detail.